Good afternoon, everyone. Welcome to the Inventor Showcase presented by the Center for Clinical and Translational Science. I'm Jan Weisenberger. I'm the Senior Associate Vice President for Research at Ohio State University, and we are a proud partner with the Center for Clinical and Translational Science in helping to present some of the most outstanding innovation work that is being done by our faculty and our partners at Nationwide Children's Hospital. One of the things that truly is important about the entrepreneurship and innovation process is, as Dr. Becky Jackson, who is the director of the CCTS, likes to say, that entrepreneurship and innovation is an important pathway to translation. It is the outcome, it's the relevance, it's the why of much of our research activity is to translate what we do into services, products, and new knowledge that can benefit society. This has been nowhere more evident than in the outstanding work that our faculty have done over the last 6 months during the COVID-19 pandemic. I have been incredibly gratified by the way that our faculty, our students, and our staff have pivoted their research focus to address one of the most important crises of our time. We have seen teams form, work being done at a pace and with an urgency that I think we have never seen before. And you'll certainly see this today in the team that is going to be represented at today's Inventor Showcase. So today's program includes a presentation called A Breath Test for COVID-19. The Inventor team is Parina Guma from the College of Engineering, Andrew Bowman from the College of Veterinary Medicine, and Matthew Exline from the College of Medicine. That will be followed by the resource team section of the program, which today is going to be represented by Carolyn Crisafulli from Ohio State Advance, Michelle Bright from the Clinical Trials Management Office, and Ryan Haley from Ambulatory Services at the Wexner Medical Center. But let's talk first about the innovation team that's going to be presenting today. This team is led by Dr. Parina Guma. Parina is the Edward Orton Chair in Ceramic Engineering at Ohio State University. She joined us just a few years ago. Her previous appointment was with the Institute of Predictive Performance Methodologies and the Materials Science and Engineering Department at the University of Texas, Arlington. Before that, she was a professor at the State University of New York in Stony Brook and was the founder and director of the Center for Nanomaterials and Sensor Development. Dr. Guma holds a bachelor's degree in applied physics from the Aristotelian University in Thessaloniki, Greece, a Master of Science in Engineering in Materials, and a Master of Philosophy in Organizational Management, both from the University of Liverpool. And her PhD is in Materials Science and Engineering from the University of Birmingham in the United Kingdom. So she clearly brings an international focus to her background and to her work here at Ohio State. Dr. Guma's research activities involve the synthesis and characterization of nanomaterials for bio and chemical sensors and for biotechnology. The way that she applies this work most recently is working in the development of personalized diagnostic breath analyzers. Dr. Guma's work here involves extensive efforts to create non-invasive breath and skin-based diagnostic tools. But she's a multifaceted researcher. Other areas that she has worked on are in the development of photocatalysts and sorbents for addressing petroleum pollutants, and then also a program that I really find interesting, self-supported photocatalytic blankets that float on water and create energy. I would say that Dr. Guma truly epitomizes the scientist inventor. She has more than 150 publications and holds 18 US and international patents. So I think she is exactly the kind of researcher that we are very happy has pivoted her work to address the global pandemic, and ways that we might move forward to do better diagnostic testing. I'd like now to turn everything over to Dr. Parina Guma.
Thank you very much, Dr. Weisenberger, and thank you, Tanya Matthews and the team for inviting us, our team of co-inventors of the breath test for COVID-19, to present our work and our journey towards fighting this terrible pandemic. And I wish to thank not only my research group, but all the research groups and everybody involved in carrying out the studies that we're going to give you a summary of today. Along with my co-inventors, Dr. Adriu Baumann and Dr. Matthew Exline. So as you're going to see, first I would like to give you an overview in the next slide of what's the background, how did we get really to make a breathalyzer? And that has been a long time in the making, almost 20 years, I will say. I have been working on this technology. Here you see a glimpse from the National Science Foundation video made on one of the early devices when you see someone exhaling and then getting a reading of the gases that are in the breath. And over the years, as you see, again, the next slide, we have been featured in many different social media, and there has been a lot of excitement, the same excitement that we have about the revolutionizing medical diagnostics by using this type of resistive semiconducting sensors that are very inexpensive, very affordable, yet they're very scientific in the way that we use them to correlate what gases are, what biomarkers are that signal disease and metabolic malfunction in breath, and making diagnostic tools from them. So next, I would like to point out that this is not new. We didn't come up with the idea of breath analysis. That was known since the antiquity. And but it was really in the 1970s when the renowned chemist Linus Pauling started sampling the breath to identify chemical compounds. And although we knew how many, about 1,000 different species, and some of them are more prominent than the others, it took until really 2000 before we started thinking of breath analysis as something viable, because there were no materials and there were no devices, no sensors that could really detect these biomarkers, these gaseous components in breath. So next, you have a list from the literature that shows you what type of chemicals we target, what type of chemicals we work with. And some of them stand out, like isoprene, acetone, ammonia. These are some of the ones you found in the highest concentration, and they are known to be related to medical conditions. And then there are inorganic compounds like nitric oxide, which is a marker for oxidative stress. But what you notice from this is that their concentration is very, very, very low. It's parts per billion in most cases. These are almost trace concentrations of these compounds. So you need to have materials that are extremely sensitive and they are extremely selective to what you want to measure. Rather, you are really looking for a needle in a haystack here. And that's where we have made the breakthrough contributions to sensing. And next, you are going to see what was really our approach towards this diagnostic breathalyzer is a crystallochemical approach to selective chemosensing. That means we make materials and we change we modify their structural characteristics. So these materials obtain high affinity for a particular chemical, for a particular gaseous component, or a family of gases. So we use the perovskites, like gamma tungsten trioxide, to selectively detect nitric oxide in breath, or we use the alpha phase of molybdenum trioxide to specifically detect ammonia in breath, and so on and so forth. And we have different mechanisms that are employed every time in every situation. And the advantage of our approach is we know exactly what we measure every time we sample the breath. And this is really a major difference between our technology and other technologies, so utilizing non-selective sensors. So the key to our approach is gas specificity. And as you see in the next slide, if we can move there, you're just gonna see the material science at play here, when you can make the same family, the same composition in many different configurations, not only in terms of size, but in terms of crystal structure, from the ferroelectric polymorphs of tungsten trioxide to cube-like faces, to hexagonal nano rods, nano crystals, and nano wires, to salt gel processed nanoparticles. Each of them have different properties that we utilize to make different sensors. And as you see next, what we really do, how the breathalyzer works, is you capture a single exhale. In a single breath, there are like thousands compounds, and some are known, some are not known, but we target a specific one, let's say ammonia. And then not only we detect this, but we are able to quantify the concentration. So now we know, is it really the concentration that is going to indicate a renal disease or H. pylori presence or so forth. And to illustrate that better, we have been using a schematic, which you're gonna see next. So you have the structure of the alpha molecular oxide, which has weak van der Waals forces between molybdenum and oxygen. And the blue and gray are the ammonia molecules that react specifically with this loose oxygen, undergo a reaction and create dinitrogen. In this way, we have very high affinity for ammonia as opposed to every other chemical that can be in the breath. So this is the principle of this selective breathalyzer. And if you see in the next slide, we can modify even the same material, change the structure, and now we go from ammonia to detect methanol. So now we play with the crystal structure, we play with these interactions, and we modify the sensor response. And all of this has been highlighted in the next slide, that is in a publication that shows really the path to making this diagnostic breathalyzer from the beginning. And there is a table in the next slide that shows what's the timeline really, how long it took us to get where we are today. So I came up with the idea and I proved the first selective ammonia sensor back in 2002, but that was for an automotive application. It was supposed to monitor urea in the exhaust of an automobile. Of course, these sensors don't last for 10 years, 100,000 miles. So, but they were proper for using them in a similar complex environment like the human breath. So the concept of the portable handheld single breath analyzer was introduced by me in 2004 for urea, asthma, and lung cancer. And from there on, we have been working on publishing and patenting a lot of different sensors, a lot of different technologies. And most important mention is 2015, when we start thinking of why don't we use 3 selective sensors to identify not a single disease, but now a complex disease like an infectious disease. And from there on, if you see the next slide, the idea was that everybody can give a single exhale. Everybody breathes, every living creature. And you're going to hear from Dr. Bowman his animal studies and from Dr. Exline on the human clinical testing. And so the essence of this technology is we sample one single breath, one single exhale. So the next innovation and the next breakthrough has been how to interface these sensors, these inexpensive semiconducting metal oxide entities, probes, with microelectronics. How can we make devices that are handheld, portable, and they are easy to use, and they have high stability, the low cost, rapid response, reversible response, and most important, how they can just sample a single breath by just having someone exhaling into a mouthpiece. And for that, I have been collaborating for a long time with Professor Stanasevich from my previous institution, Stony Brook. And the next slide, you see one of the latest prototypes we have been developed and used that shows how low concentrations of gases we can detect, low part per billion. And you can see also that it's an integrated small device IC, all microfabricated. We use a variety of sensors with baselines from 1 kilo ohm to 100 mega ohm and a very low power consumption. And the next slide shows Dr. Stanasevich, who is not an OSU person, so he's another co-inventor, but he's not in this team, and how we try to interface with mobile devices in the next generation of devices. So the next slide is 2017. The flu breathalyzer paper came out, and it created quite a stir because a lot of people and a lot of press, there were a lot of press releases, there were a lot of things that a lot of people were talking about it. So it didn't take, and they were talking about it for a long time, because is it really likely to have a breathalyzer for infectious diseases? Can you have a flu breathalyzer? And that became even more important when COVID emerged. And if we go to the next slide, please, you're going to see now the timeline from where the flu breathalyzer was made to how it came to the COVID-19 breath test that we're discussing today. And it was February 12, 2020, when we received a phone call from the White House, Dr. Navarro's office, inquiring whether the flu breathalyzer can be really used for COVID-19 detection. Now, at the same time, I have been interacting with Dr. Baumann, who is an expert in infectious diseases and has been studying them in animals. And we had started thinking about animal breath testing for flu. So then we brainstormed what will be the likely biomarkers for COVID-19. And soon afterwards, we reached out to Dr. X-Line, who has firsthand experience with treating COVID-19 patients. And he was also very positive in joining the team and help us develop a bedside test for COVID-19. Our team collectively applied to the National Science Foundation for funding under the Rapid Year Response to COVID-19. We received the maximum amount they offered, $200,000. And with that, I'm going to pass it over to Dr. Andrew Baumann, who's going to discuss, give you some more details about these studies. Thank you. Andy. And
yeah, so there we go. So yeah, we started looking at how we would use the breathalyzer technology.
yes, so I think we have a problem with the connection with Dr. Baumann because being the amazing scientist that he is, he's on the field carrying out research at the moment. So I think what we are, until he's able to get reconnected, I'm gonna show you what we have been really doing by having the student demonstrating how the testing has been happening. When we use this we have been using a 3-sensor array device, and we have been collecting bags, single or 2 exhales in these 1 liter bags. And the next slide, the next slide we're gonna see that our project is really using a single exhale and then taking it within 15 seconds, we can get a response. Now there is this urgent need for reliable, accessible, rapid diagnostics. And that's the need we really respond to. And the principle of COVID-19 sensing can be really transferred to other infectious diseases and other health crises, new strains of flu, or to COVID, or even to Ebola. So the next slide is showing you the setup that we have in Dr. Baumann's lab, and he was gonna explain much better than I do. But here you see the biosafety cabinet with, we take the samples like Dr. X-Line is gonna discuss next, and we sample them in the single sensor device that is down there, a very, very small thing. And we use these chemoselective sensors to detect the biomarkers of COVID-19. And we really try to make it a diagnostic as opposed to a screening tool. And that's something to hear from the other colleagues on this showcase. We offer gas selective sensors for breath biomarker detection. And currently we're exploring rapid manufacturing and deployment, as well as regulatory approval. With this, I would like to pass it on to Dr. Exline. And again, thank you for your attention.
We were very excited about this technology at the medical center. To date, we've had almost or a little over 1,200 admissions with COVID-19. Roughly 40 to 50% of those patients end up in our ICU. And at our peak, we had about 140 patients with about 50% of them being in the ICU. So the COVID pandemic definitely hit our system hard. And because of this, we had to greatly expand our ICU services, and any device that would allow us to lessen the burden on our staff and our patients was obviously very exciting. We needed to put the device to the test, and we were trying to think of where we could start. We needed to know that we had a defined group of positive patients. We also needed the appropriate test negative controls to prove we could differentiate those with COVID-19 versus those that do not have it. And then it would be ideal if we could see these subjects back on a repeated basis so that we could follow the trajectory of their disease and see kind of what our window of detection is. And so we went into the ICU environment looking to see if that would be a good ideal testing ground. The first thing we had to do, though, is think about with COVID-19, how do we keep the staff safe? So the quirk to research in our current environment is it's about safety of the staff first. So how do we preserve PPE? How do we make sure we're not exposing everyone? So we had to figure out a way to bring the test to the patient without taking the device into every room and potentially contaminating it. We also needed to make sure we were sending the samples to the lab with as low a risk of infection as possible. So we came up with a device where we use a viral filter on the exhalation circuit of a patient's ventilator. We then connect the breath bag onto this exhalation circuit. Our respiratory therapist then adapts the bag, the tubing into a breath bag, and we collect several breaths worth of exhalation and then take it to the lab. And one of the things you might notice is that this person is not wearing a full gown and gloves. These pictures were in a non-COVID room. Do not worry, we always followed appropriate PPE. So far, we've collected 27 patients and counting. Our goal is to get up to 46 total. 18 of our patients have been COVID negative, so they've had a test and been negative. These are our standard ICU patients. And then we've had 9 positive COVIDs. You can see the COVID positive patients, despite what you might think, were actually slightly younger than our standard ICU patients. We use this ratio called the PF ratio. This is a measure of how well they're oxygenating on the ventilator. And what we see is our COVID-19 patients had lower oxygen settings, as you might expect. And this correlates with a moderate to severe form of acute respiratory distress syndrome. So what we found in our first patients is we are seeing a pattern of detection in COVID patients that differentiates them from the control. We call this the omega pattern. Overall, on the first day, we see the majority of our COVID patients are showing that characteristic pattern, whereas on our controls, the vast majority are not showing us that omega pattern. A few of our control patients did have the COVID pattern. These patients tended to be those with other respiratory infections. And then most exciting to us, as we might expect, as these patients improve over time, they look less like COVID patients and more like standard respiratory failure patients. So that was very exciting to us and gave us proof of concept, but I think the use case will eventually be using it in an ambulatory setting. So we went to our swab stations. As you all know, the medical center has been testing patients throughout Central Ohio and featuring these drive-up swab stations. And we think this is where we can really make our use case. We also needed to get our team together. And so this is our first day going to the swab station. And this is Sarah and Emily, our research coordinators, who have done most of this work. And we really couldn't have done it without our partners in the CTMO. And that's highlighted by Michelle, who is the manager of the CTMO and really guided us through this process.
Great, thank you, Dr. Exline, and hello, Inventor community. As Dr. Exline said, my name's Michelle Bright, and I'm an assistant director in the Clinical Trials Management Organization. I'd like to go over with you today just a brief overview of what the CTMO is, and then also how we've been involved with COVID-19 research. So this next slide will just give you a little bit of information about the purpose and goals of the CTMO. CTMO was initially formed back in 2011 with a purpose to provide administrative infrastructure for the management of non-cancer clinical trials in partnership with investigators and their respective departments or divisions. Our primary organizational goals include things such as helping to strategically align clinical research resources and personnel, increase compliance and decrease risk, and this can be in areas such as regulatory, patient safety, and even financial. Identify barriers and develop solutions to decrease timelines to study startup and improve enrollment, and also provide comprehensive reporting at the division, department, and college levels. Bottom line is we're here to help the investigators navigate the complex landscape that research can present at an academic institution. Our structure on this slide is both centralized and decentralized. So on the left side of the slide, you see the leadership of our centralized part of CTMO. And that's our medical director, Dr. Brad Rovin, our senior director, Dr. Deanna Goldenkreutz. And then we have 3 offices, the finance office, directed by Sandra Serafin, our integrity and regulatory compliance office, which is directed by Brooke McDaniel, and our operations and implementation office, which I am part of, is directed by Todd Lush. Within the CTMO centralized structure, we have upwards of 35 staff members consisting of regulatory compliance officers, research budget analysts, clinical research managers, and other research staff. On the right side of the slide, you'll see what kind of represents our decentralized part of, and it's kind of small, I realize, so I'll just summarize what's happening over there. But these are the different departments, divisions, and centers and institutes that we work with. We're in 9 different departments, over 64 distinct research sections and divisions. We also work with 9 centers and institutes, as well as the Clinical Research Center. Next slide gives you a snapshot of what the CTMO provides for their investigators, everything from protocol feasibility, regulatory management, which includes IRB submission, executing CDAs and contracts, creating comprehensive study budgets, managing the finances from start to finish through a trial, overseeing compliance, and we're involved in the development and maintenance of standard operating procedures. We work closely with the study team. It's one of my main roles, work close with the study team on training and oversight for, again, compliance and helping them implement studies. And then overall study management, which can be anything from protocol implementation to recruitment and enrollment strategies through study closure, or even helping the groups through inspections or audits. The next couple slides are going to show you, it's more specific, what our involvement has been with COVID-19 research. The CTMO has been very involved with COVID-19 research from the start of the pandemic. Our leadership worked closely alongside the Wexner Medical Center, the the Comprehensive Cancer Center as well as the College of Medicine to help create and develop the current COVID-19 research policy, which you see there on the screen. This policy is in place to help ensure that research moves forward carefully. As Dr. Exline mentioned, first and foremost, patient safety, as well as hospital resources were taken into account. And we also want to make sure the research is being done compliantly and in an organized fashion. As part of this policy, the investigators have to submit their research proposals through a REDCap survey process to be reviewed and approved. And Drs. Robin and Goldenkreuz serve as reviewers for the non-oncology, translational, and clinical research proposals. To date, there's been well over 100 proposals submitted, and with more coming in, every day. Of course, there's ongoing COVID research happening. CTMO also worked with the College of Medicine in forming working groups. The purpose of these working groups, we found early on that there were lots of people that wanted to be involved in COVID-19 research, different divisions, different departments, different disciplines. The working groups are a way to bring people together to streamline and work towards one common goal. It was to discuss trial feasibility, discuss screening and enrollment strategies to ensure that all clinical trials that we took on, all research that we were doing would be equally successful. The three main working groups that I personally have been involved with are the inpatient, outpatient, and the biorepository and healthcare worker. And that's with the inpatient is where I've been working with the critical care team on this particular project. With the next slide, the biggest part that CTMO has been involved with COVID-19 research is helping to implement the clinical trials. So once they've gone through the approval process and are ready to be implemented, we've worked closely with our partners, our Office of Research, or Office of Regulatory, I should say, has helped streamline the IRB and IBC submissions. So it's maintaining close communication with them to make sure that we were keeping up with all the changing rules and regulations and still doing things compliantly and in an expedited fashion. Our Office of Finance helped with fast-tracking contracts and budgets, getting things executed quickly. A lot of the COVID-19 research has very short timelines. Obviously, we want to get the research done and get the results out there quickly. Our Office of Operations worked closely with the study teams, and this is where I came in working with Dr. Exline and Sarah and Emily closely on finding ways to adapt our current processes. So adapting the consent process and the study procedures to again ensure staff safety, decrease exposure. We had to come up with ways to do remote consenting, electronic signatures. In the case of this particular project, we got approval for waiver of consent documentation to streamline the process even more and also continue to decrease staff exposure. CTMO worked to help connect shared resources and expertise. This was largely through those working groups. No need to reinvent the wheel. If one group had figured out a way to do something, get around some hurdle with COVID-19 research, then we shared that information to help everyone move forward. And then lastly, collaborating with our clinical care units. That was a big thing CTMO helped to facilitate as well. None of this COVID-19 research would be successful without the support of the clinical side. The nurses have been an integral part. Respiratory therapists obviously played a big role in this particular project. And then our next speaker, Ryan, with ambulatory services, played a big part as well. Very integral partner, and we wouldn't have been successful without them. So I'll turn this over to Ryan to tell us a little bit about ambulatory services.
Thanks, Michelle. Thanks for turning it over. Like she said, I'm Ryan Haley. I'm the Senior Director for Ambulatory Services. Before COVID turned our world sideways, my day job consisted of managing our outpatient clinics in ambulatory, our primary care, and our orthopedic and sports medicine practices. And so when I took on the job of running the swab stations in July, I have to give a lot of credit to the emergency department that set it up at the onset of the pandemic and handed it over to me. When Matt Exline reached out to me and asked about this exciting invention and project, I was super stoked about this because I thought if this thing could actually put us out of business at the swab station, I'm all for it. So Let me kind of take you into a little bit of a glimpse in our world and what we do at our quick test stations, affectionately known as our COVID swab stations. So as you can see here on the visual, we actually do a little bit more than just COVID testing. Because of the aerosolizing nature of a COVID test, you know, those things have to be done outdoors. But what many of you might not know is that we also have to do our flu and strep testing outdoors. So we have that set up as well at our quick test station. We also are equipped to do blood draws for patients who are COVID positive or in a rule out COVID status, so to not kind of track unnecessary COVID positive patients through our ambulatory sites when we have an alternative solution. Additionally, we're excited, we haven't launched this fourth icon yet, the upper respiratory care clinic. We're starting that in the fall, actually in just 2 weeks, so that our providers who are taking care of patients via telehealth have an opportunity to refer patients there who may be COVID positive and need to be seen and evaluated in a safer environment. So we're really excited about that. And lastly, we just launched last week drive-through flu vaccines. We do these in off hours so that we can not kind of intermingle the patients who are coming through for flu shots with our other symptomatic patients. So as you can see, a lot of different activities at the swab quick test stations. Next slide. But you all came here to kind of listen to what we're doing with this new invention, this breathalyzer invention, as well as our swab operations. And just to give you a little bit of history, since we started the operations in the outpatient setting at Ohio State, we've swabbed over 83,000 people and counting. So really impressive amount of volume that has come through our outpatient settings through the outdoor drive-through sites. We were at kind of the peak of this in July when Priority 5 testing was open and anybody that wanted a test could get one. That has since died down, so the days of 800 plus are a little bit in our rear view, thankfully. I can honestly say that this is one of the proudest times of my career, not because of just the work we're doing here, but just the way everybody's pitched in from the medical center. You know, I've had amazing volunteers across the system, from RNs to physical therapists to athletic trainers to doctors to nurses. It was a really hard effort to recruit people out of their day jobs to come help us swab. I like to joke that every day I felt like I was coordinating a pickup game of basketball to assemble our crew of swabbers to swab the symptomatic patients in the city of Columbus. And then, as you'll see, we had to face a lot of challenges from weather and nature. You know, it doesn't stop. So providing medical care outdoors is something new for us. I always like to say a good day at the swab station was a day you didn't have to yell, put the car in neutral, or chase a goose away from the testing site with a broom. So those things happened multiple times. I had to do both. And then pretty crazy that you'll see people coming up in all sorts of different vehicles, from bird scooters to hoverboards to bicycles to Ubers. So, but I think the last item, the 0 there, is what I'm most proud of, is the fact that we've really kept it safe for our staff and our employees, that of all those thousands of patients who've been swabbed, there's 0 confirmed transfers of COVID-19 to any of our staff members. Just a quick walkthrough. The process is simple. That's the only way we're able to do such mass amounts of testing. The COVID tests are ordered in the clinic by the providers or through our call center if they don't have an OSU provider. And the patient, all they have to do is drive up to the testing site with a piece of paper on their dash as their name and date of birth. We're also able, during the breathalyzer study, to actually indicate those who are participating by putting a different color sheet of paper under their windshield wiper. You know, we use tons of visual management outside because we want to reduce the amount of opening and closing of windows and conversations that we're having. So we try to make it efficient without having to do all that. Once the patients get their labels placed under their windshield wipers, they drive up to the next station and they get their nasopharyngeal COVID test performed. And so at the swab stations, as you can see pictured there, when we were doing this study with Matt and the team, we were also having the swabbers then collect the breathalyzer samples. So all of that was done at the same site. Amazingly, patients were very eager to contribute to this kind of scientific breakthrough, and we're super cooperative. And so as you can see here, here's the 5 locations we have. It's pretty incredible. We have got sites all over town, from Ackerman to our East location. We support our Columbus Public Health partners on Tuesdays and Fridays. Student health with our campus constituents back for the fall. We're doing testing every day at the Wills Center. And then, of course, our OSU athletics. Thankfully, we've got a plan in place to have football back for the fall, and we've got a really great testing plan that we're supporting them with. Next slide. So this last bit of mine is that you know, life and swab life never stops. Once you get settled, it's time to change. So we've moved sites, we've had to change things up. And the really kind of exciting plans for the fall is that we're about a month away from moving the operations at our Ackerman site, our main hub that does the most volume, as well as our Columbus Public Health location, over to the Expo Center, the Ohio State Fairgrounds. And you can see the facility there on the right is the Celeste Center. We plan on having cars drive through. You can see a floor plan there in the middle, vertically through the facility, and do the registration and testing indoors. And why this is important is that as temperatures drop into the low teens and sometimes lower, it's going to become really difficult to perform this in a safe and effective way. I mean, staff conditions are going to get pretty brutal. So being able to do this in a ventilated environment that's covered is a clutch win for us. So we'll be partnered up there with Ohio Health, Mount Carmel, and Ohio State, as well as the Columbus Public Health Department to do this 6 days a week. And so it's going to be a great thing for our staff, and we'll also get some economies of scale and efficiencies by bringing some of this together. So yeah, thank you. And next slide. So just really quick, just if there's any future research opportunities that you're looking forward to reaching out to me about setting something up at our swab locations for help, there's my information. Please feel free to reach out to me. And I will go ahead and pass the baton over to Caroline Crisafulli. Thank you.
Thanks, Ryan. As Ryan mentioned, my name is Carolyn Crisafulli. I'm the entrepreneur in residence for the CCTS Optimizing Innovation, Commercialization, and Entrepreneurship Program, and the Director of Innovation for Ohio State Advance in the Office of Research. Next slide. Ohio State Advance launched in 2008 under the auspices of the grant from the National Science Foundation's Advance program to recruit, retain, and advance women faculty in STEM. Our signature program, Reach for Commercialization, launched in 2010. Reach is a year-round resource and network for women interested in amplifying the impact of their research by exploring technology transfer. Next. Our program is dedicated to narrowing the persistent gender gap in tech transfer, patenting, and entrepreneurial pursuits. This graph from a 2020 report released by the USPTO shows the participation rate of women in patenting. Although 53% of PhDs were awarded to women in 2019, the percentage of unique women inventors listed on US patents is still less than 13%. Next slide. Similarly, a recent report by the Diana Project shows that only 15% of all venture-backed startups have a woman on the executive team, and less than 3% of venture-funded companies have a female CEO. Next slide. The REACH program paints a broad overview of the technology transfer and commercialization process presented in 4 quarterly core themes: visioning the impact of research, learning the landscape, building a team, and understanding the funding life cycle. In addition, in-depth small group workshops are offered throughout the year to dive into greater detail on the core topics, as well as individualized mentoring and counseling. Next slide. One of the most important aspects of REACH, excuse me, is the network of accessible near peers, role models, mentors, and resources. We welcome women from all disciplines across the university, including faculty, staff, postdocs, and grad students. The diverse interdisciplinary network results in a thriving community of professional women with a wide variety of domain expertise and life experiences. Next slide. Just over 100 women have participated in REACH since its inception. REACH alumni are associated with more than 300 invention disclosures, nearly 100 issued patents, and 9 startups. Next slide. And final slide. We welcome all women who are interested in increasing the visibility and impact of their research and want to expand their professional network. Please feel free to reach out with any questions. You can reach me by email. I'm crisafoli.1. And with this, thank you for joining us today. I'd like to turn this over to Bruce Weinberg for the Q&A session.
Carolyn, thanks so much for, for the introduction. I'm Bruce Weinberg, and I'm a professor of economics and also the program director for the Innovation and Entrepreneurship Group at the CCTS. So I'm here to welcome folks back to the screen, all of our panelists and presenters, and to moderate the Q&A session. I saw that there was already one question that was posed and then answered as part of the Q&A function, but please feel free to post additional questions there as well. So I wanted to start with a question or two that I had for Parina. So I guess one question is how long do we think it's going to be before the breath test will be available, and then how does this relate to the advantages and disadvantages of other COVID-19 tests?
Thank you for the question. So we are now in talks with the FDA and in the process of seeking regulatory approval. And it's the use case that I guess my collaborator, Dr. Exline, can discuss more with you. So we are very hopeful it can be soon. So Matt, if you'd like to add to that.
Yeah, I think we're very excited about it. We are concluding both our ICU study and also our what we call high throughput study, which is the swab station study that Ryan is helping us with. And as soon as we get that data analyzed, then I think we'll be ready to see how we can use it. As Parina said, our main goal has never been that we can get 100% of COVID cases. Right now, Ryan's having to swab hundreds of patients a day. If we could take that number down and make it tens of patients a day, our positivity rate on the swabs would go up, but the number of people who have to go through an unpleasant procedure and then sit in isolation for 2 days waiting for their swab results to come would be greatly reduced. And so I think that's kind of the use case we're seeing.
That's the fastest one that we envision, the first one.
Just a moment.
Great, thank you. Again, please feel free to post questions to the Q&A. We do have a few minutes for them. So I guess for Michelle, one question is, how is all of the COVID activity affecting non-COVID activity? And are there ways of maintaining non-COVID activity during the current environment?
Yes, thank you for that question. That has been a challenge, certainly during the initial kind of March, April, May, even into June, when most non-COVID research was either on hold or had at least slowed down. We certainly were still doing research, but had to find new ways to do it. We could focus a lot on the COVID research, but then as research started to reopen or resume and a lot of the hospital procedures and activities started to resume and all activities increased, that's something all the groups are, a challenge all the groups are facing. Some of the groups kind of overnight, their workload it doubled, went from maybe 5 trials to 10 trials. So that's something, at least in the groups that the CTMO works with, we're trying to help navigate that. Again, use a lot of shared resources, not reinventing the wheel. Like I said, if another group's already come up with a way to do something, we're trying to share that information to decrease the workload. But it is a balancing act to still keep the non-COVID research happening, and not the way it was pre-COVID, where we have to do it in a way that is, again, still keeping the staff safe and decreasing exposure. So it certainly has been a challenge and has kept us on our toes. We're learning new stuff every day.
I guess we're all learning new stuff across the university and across all of life at this point. There are a number of questions that have been posted to the Q&A, so I don't know if people want to jump on one or more of them. Ryan, I was interested in if you had thoughts on sort of what the future of testing for COVID would be. I mean, it's quite an impressive operation that you've set up and dealt with quite a range of circumstances.
Yeah, I mean, I think we hope that it can be more efficient and as Matt alluded to, quicker turnaround times. It's incredibly expensive to set up a swab station. That operation's costly from just staff resources, the tent, and it's a very crazy environment. I'd have my wife bring me a change of clothes one day because there was a thunderstorm and we were soaked out there telling lines of cars that we were on a 30-minute lightning delay. So it's not the environment you want to work in. So we always like to joke there's swab station bingo and there's a lot of different items on that card that would raise some eyebrows. So we would be eager to find a solution to put us out of business pretty soon here.
Well, hopefully we've got one at our fingertips soon. Do people, I don't know, I think many of these questions would be for Parina that are coming through the chat, but do you want to try to, or anyone want to try to respond to any of them?
That's fine. So one of them is how do the COVID markers differentiate with the non-COVID respiratory diseases, such as seasonal flu? We have different biomarkers for COVID and different biomarkers for flu. That's all I can comment on now. So I hope that answers the question. Are there any other breathalyzers approved or used internationally? No, there are not. And then what I kind of read out the breathalyzer gives is yes or no. For the time being, we get patterns, but yes or no. But I guess Matt can also comment on.
Oh, no, I think you covered it. I will say from a lung doctor standpoint, a lung physician standpoint, we use a breathalyzer, actually, as Parina alluded to at the beginning, to look for asthma activity. And so there is some opportunity that we've used devices like this in the past, but if we could expand it into a more rapid fashion for a disease that patients want the answer. I think the one thing we're underselling in our current testing environment is the economic cost of how we test now. We ask everyone while they're waiting for their test to isolate, which is 1 to 2 days off of work, 1 to 2 days away from their family, 1 to 2 days where maybe they defer on doing a medical procedure because they don't have that amount of time to isolate themselves. So that's a huge cost to our system, to say nothing of, until I worked with Ryan, I had no idea how much those tents in the parking lot cost to rent. So if we could find a way to reduce that cost, it's a huge win, both for the medical system, but then also for our patients.
Thank you. It looks like Parina is responding to some more questions, which is great. We had a question while we were doing this about setting up the series about how this the COVID-19 pandemic has affected all of you who are participating. So I'm going to launch a poll and ask people to respond to it. And then while we're doing that, I just wanted to thank all of our speakers. I think this is both for the fantastic presentations and for all the wonderful work that you've been doing. It's been a great talk and wonderful hearing what you've all been doing to address this. It does seem like just watching the poll that this has been considerably disruptive to folks. So in addition to thanking all of you, Parina, Matt, Ryan, Michelle, Carolyn, I also wanted to let folks know that that we have a new series coming up in October, in a month. and that's going to be Chunji Zhang and Yijiao Dong, who are going to be talking about constructing a COVID-19 vaccine on October 20th at the same time. In addition, we'll have an opportunity to register for that. And then I want to obviously thank the CCTS, Nationwide Children's Hospital, and the Office of Technology Commercialization, all of our partners for supporting this activity. And to all of you, thanks for coming and making this such a rich discussion today.